Okay, so now we're just going to wrap it up looking at a few other types of non-Mendelian inheritance. Again, non-Mendelian inheritance means that there's something different than just two alleles for each gene, and the alleles are dominant or recessive to each other. So one example is codominant. Codominant, much like we already learned about with multiple alleles, codominant means that they are both expressed. Okay, so if you have white camellia flower times a red camellia flower, you don't get offspring that are white or red, you get offspring that are white and red. They are both expressed. So these cells are expressing the white phenotype, and these cells are expressing the red phenotype. So you get kind of a splattering, I guess, where they're both expressed and somewhat equally. So if we're going to cross a heterozygous red and white times a heterozygous red and white, if we were going to cross a red and white camellia with another red and white camellia, we would get some red, we would get some mixed red and white, and we would get some white. Oh, and we would get them in ratios, right? So 50% would be red and white, and 25% would be red, and 25% would be white. What about if the traits are mixed? Okay, so you get an intermediate phenotype. This is a little different. So in snapdragon flowers, and I know the Amoeba Sisters video talks about this, same letters here for red and white, but a red times a white is going to give a pink, not a red and a white. So same setup for the Punnett square here, and we're going to get, if we cross a hybrid, we'll get 25% red, we'll get 25% white, and we'll get 25% pink. All right, another concept we'll spend a little more time on, epistasis, is when you have interaction between genes. So for instance, this is what happens in labs, Labrador retrievers. This is really what's going on. So, you can have black lab, chocolate lab, so like a browner colored lab, and a yellow lab. And the yellow labs have different shades, again, depending on other factors. We're not going to go into that. We're just going to say it's either black or brown coat or yellow lab. Okay, so we have these genes that are capital B, black coat, and lowercase b, it's brown coat. But the thing is these will not be expressed unless you get this capital E gene as well. So if you don't have the capital E gene, then that's not going to do anything, and it's going to be a yellow lab. Okay, so what if we had, so we'll do this cross, what if we had a black lab? And it's, again, how do you know it's a black lab? Because it has the capital B, and it also has an uppercase E. Remember, the uppercase E will help that be expressed. So what if we had a black lab that is heterozygous for the black coat and heterozygous for the E gene, which helps the expression of the black coat, times another one. So again, do the FOIL method. First, outer, inner, last. And then it's going to be the same on both sides. And we will have to fill out this whole thing because it's pretty unique. And then we'll take a look and see what the phenotypes are. Okay. All right, so I guess I'll use that for black labs. So how many of these are black labs? That one is, because it has, you have to have a capital B and you have to have a capital E. Yes, yes. So 9/16 are black labs. And then how many are chocolate? Chocolate would be a lowercase b's, and also would be lowercase, no, so lowercase b's and one uppercase e or more. That doesn't really show up very well. I'll try to use a different color. I guess that's it. So chocolate. Okay, those are, so 3/16 brown, and then the rest are yellow labs because they don't have the uppercase E, so the code color isn't expressed. So yellow labs for 4 of those. So similar to a two-trait cross that we've been looking at, but different because epistasis, right? We don't get a 9:3:3:1 ratio for anything. We get a 9:3:4 ratio. All right, last couple of topics. Hybrid vigor, I'm just going to gloss over. This happens in animals and plants. It's where if you take, if both of these are purebred for certain traits and they're crossed, and the result of the cross is the middle one is a hybrid. And what happens is you get these plants or animals or other organisms that have traits that are outside of the norm for the purebreds, and they call that hybrid vigor. And so we use that in agriculture a lot, and we seed companies will come up with these pure plants that would not be very good for like consumption, but the seeds of the pure plants are then used to develop seeds for hybrid plants, and then that's what farmers will grow, is they'll grow the hybrid seeds, and the hybrid seeds exhibit something called hybrid vigor, which means that there's going to be a lot of kernels, it grows faster, all sorts of things like that. All right, and then another thing that can happen is in genetics is that sometimes you get traits expressed dependent on environmental factors. So classic example is reptile eggs. And so like if a painted turtle lays its eggs in the springtime and it's a really cold spring, so that's example 2. If it's a really cold spring, the temperature would be here, and then you'd have a lot of males. And if it were a really warm spring, it'd be here, and you'd have very few males. So like at certain temperatures, genes are activated or inactivated, and those genes that are determining gender are activated or inactivated. And a lot of actual animals go through even gender differences as their lifetimes. Like a classic example would be like clownfish. Juvenile clownfish are neutral gender, and the dominant clownfish in a clownfish harem is female, and the subdominants are males. And if the dominant female dies, passes away, leaves, then the most dominant subdominant male actually reverts gender to female and grows larger. So there's lots of environmental factors actually that determine different traits. And the alligator example is similar to the turtle one, except that there's a range. So you get this males being expressed both at cold temperatures and warm temperatures, and females being expressed in intermediate temperatures. So go ahead and try to do this problem if you'd like to. It's an example of a AP Bio question that is related to this concept of environmental impact on gene expression. I guess I'll do it here. In this fish, I can't even say that, Cynoglossus semilevius. Sex is determined by a combination of genetics and environmental temperature. So male fish that are genetically male have 2 Z chromosomes. So I'm just going to write this down here. That's genetic male. Okay. And genetically female fish have 1 Z chromosome and 1 W chromosome. So I'll move this here. Okay. When fish are raised at 22 degrees, ZZ fish develop into males, and ZW fish develop into females. So that's kind of this. So if that's what their genotype is, that's what their phenotype will be if they're at that temperature. Okay. However, when fish are raised at warmer temperatures, the Z chromosome is modified into a Z asterisk. Okay, Z asterisk W fish develop into phenotypic males. So at 28 degrees, we get this happening. And they're also males, but they're genetic female, but they are phenotypic males. Okay, so that's a genetic male, that's a genetic female. A genetic female can develop into a phenotypic male. This chromosome gets modified. Okay, and they could pass on the Z asterisk chromosome to their offspring even when the offspring are raised at a different temperature. So that modified chromosome can be passed on to offspring. A cross between a ZW female and a Z asterisk Z male. Okay. So that's gonna, yeah, that would be male. Is shown. So, okay, so here we have 4 different outcomes. And, okay, that's male, right? That's a male as well, because it's a modified chromosome, even though it's female genotype. It's a modified Z chromosome. That's a male, so 3/4 are male, and 1/4 is female. Predict the percent of phenotypic males among the F1 offspring of the cross shown if the offspring are raised at 22 degrees. So that means since they're at 22 degrees that there aren't going to be any more modified chromosomes. And so those are my ratios. If those offspring are raised at normal temperatures, then we get 3/4 phenotypic males and 1/4 phenotypic females.